Hi everyone, it's Mr. Sinti, and today I'd like to discuss with you some of the glory of eukaryotic gene expression, and in particular, this video is going to look at transcription initiation, because as it turns out, a lot of gene regulation, in other words, the turning on or turning off of particular genes in order to enhance cell specialization, occurs at the transcription level. And so that's what this video is going to be all about. And I hope you are going to enjoy it. First thing that I want to say about this is that a little bit about how chromatin is affected. Now, you may recall from some background that DNA is wrapped around histone proteins, and that histone proteins not only help to condense the DNA, but they also help to regulate how tight it is around those histone complexes in the nucleosome. And so acetylation and methylation affect protein synthesis ultimately by affecting gene regulation. And so if you recall, acetylation and methylation is chromatin modification. But I'm still kind of discussing chromatin modification a little bit when I talk about transcription, because it does affect the chromatin structure. So this particular video is going to look at transcription. And so just a brief before we get into any detail. The thing about the detail is that, you know, the further you walk metaphorically on a hike, or the higher you climb, the better the view. But you have to sort of have a bit of a map ahead of time. And so just to sort of back it down just a little bit, you know, we're just talking about DNA as residing in the nucleus in a eukaryotic cell. And one of the most important things is that DNA is transcribed into RNA. And so literally what we're talking about is DNA being converted to ribonucleic acid. And now that process is called transcription. Transcription is mainly occurs as a result of this really important enzyme called RNA polymerase, which catalyzes the polymerization of RNA. Now, that attaches to non-coding regions upstream of the gene. Now, right over here, I'll just draw that as being the gene, right over here. In other words, the nucleotide sequence that codes for a polypeptide. Now, RNA polymerase isn't going to have an easy time attaching to the promoter, if you recall this. It's going to need additional proteins to assist it, and those are appropriately called transcription factors. And there's general transcription factors that are important in every gene, but there's some specific protein factors that I want to discuss in this video that will allow transcription initiation to be regulated. Okay, so that's what we're going to be kicking around. And so the association most associated with most eukaryotic genes are multiple control elements. So there's a few regions of DNA, segments of non-coding DNA, that serve as binding sites for transcription factors that help to regulate transcription. So there's a few of these control elements, in other words, regions that initiate transcription. And so the control elements are places where transcription factors will bind, and they're critical to the precise regulation of gene expression. And again, the importance of that, you're like, well, why should I be interested in transcription initiation and eukaryotic gene expression? Because it influences specialization within a cell. So in other words, the cell is capable of differentiating and becoming a particular type of cell. But even, I think, cooler still is that the cell needs to regulate gene expression, because it would be wasteful to produce genes when you don't need them, and it's useful to produce proteins, for example, enzymes, when you do need them. And so this is how cells can initiate transcription and ultimately regulate protein production. So to initiate transcription, The RNA polymerase requires, again, as I've been trying to emphasize, the assistance of transcription factors not shown. These are proteins, and so they're involved mainly in this area right here in the promoter. And there's general ones that are essential for the transcription of all protein coding genes. And these are protein coding genes right over here. In other words, these are the structural genes. Exon, as you recall, are the sequences of DNA that specifically code for the protein. And exons are big regions of non-coding DNA that ultimately need to be edited out during RNA splicing. Those are transcribed from the gene, but they need to be edited out at post-transcription, so RNA processing, that's referred to as. And then again, you may recall this, the RNA, since we're discussing that, a 5' guanine cap is placed onto the front of it. It helps it leave the nucleus and helps it engage the small ribosomal subunit as in the process of translation. And then also this polyadenine tail is added onto the end of the molecule, about 200 long, and again that helps the process of translation take place. Then RNA polymerase moves along past that area. The bars suggest that there's a long sequence of DNA there. It reaches a termination sequence, and then the RNA polymerase disengages with the DNA. But our emphasis today is in this area, how RNA polymerase attaches to the promoter region. And you got these areas called control elements. Do you see here? Some are in yellow. Some of them are pretty close to the promoter, and some of them are really, really far away. And so those are distal. In other words, really far away, proximal meaning close. And these areas are going to be places where proteins are going to attach to DNA, and then that's going to coil around, and it's going to help other transcription factors bond to the promoter and thus allow RNA polymerase to bind as well. And so a little complicated. And again, the hike is a little steep today, but I hope you're here to enjoy it, and I'll make it as clear as I possibly can. So in eukaryotes, the fact that there's high levels of protein expression has to do with specific transcription factors that are important in particular cells. So these transcription factors are crucial because they can locate the right gene at the right time. And so it's important. So enhancers, again, are these areas that help activators bond to those areas, and then specific transcription factors can then attach to the promoter. So they're proximal, meaning close to the gene, and some are distal, meaning that they're pretty far away from the gene. And so an activator, again, is a protein that binds to those non-coding regions, and then it sort of loops around and attaches to the promoter and allows for transcription factors to bond to activators. So it's a protein-protein interaction, and it results in the transcription of a particular gene. And so here's a picture of this. This happens to be called MyoD. It's an activator that attaches to transcription factors that helps to initiate normal protein production, myosin in particular, in normal muscle development. Let's take a look at a video at this time showing what I'm trying to describe, which is this whole process of RNA polymerase attaching to the promoter.
Most of the fine-tuning of gene expression occurs at the transcription level, making an RNA copy of a DNA sequence. Recall that to initiate transcription, an RNA polymerase must bind to the promoter region adjacent to a gene.
Okay, right there.
This cannot occur without the help of specific protein transcription factors. Some transcription factors, called activators, bind to enhancer regions some distance from the gene.
Distal.
The DNA bends, bringing the activators closer to the gene, where the activators interact with other transcription factors and RNA polymerase.
And so you can see that without those activators, you wouldn't get the transcription factors to properly bind to the promoter, and therefore RNA polymerase would have a difficult time, here it is over here, binding to the promoter itself. And so these activators provide the protein-to-protein interaction that helps RNA polymerase attach to the promoter and then move down and polymerize the gene, both exon and intron at that point. So I hope that animation was illuminating, as it often is. So some transcription factors help to, in instances, we're talking about regulation. We're not talking about catalyzation always. Sometimes the transcription factors function as a repressor inhibiting a gene from being transcribed. It's not as simple as the repressor protein, if you're familiar with this, that bonds to an operator sequence in prokaryote, but it's the same notion. There's repressor transcription factors, and then there's activators that help to either promote or silence transcription. And transcription is one of the hearts of how a gene is regulated. You either transcribe the gene or you do not transcribe the gene. But there are other ways in which gene expression is regulated, which we'll also discuss. And again, these activators attach to non-coding regions called enhancers that are distal or proximal, and then those engage activators, and then activators, with the help of mediator proteins, love that, attach to transcription factors, and then there's even a protein that helps the DNA bend over like that, and then all of that forms this transcription initiation complex, a quorum of activators and mediator proteins and transcription factors and polymerase. And polymerase itself is made up of many subunits, if you like that. And then all of that allows RNA polymerase 2 to move along, obtaining new RNA nucleotides in the nucleoplasm and begin to transcribe the gene. Yeah. Okay, so at this point, I want to sort of take our conversation after the messenger RNA is produced. There's some so that's after. There's some post-transcriptional regulation that's important here. And that transcription alone doesn't account for gene expression. It's important, but it doesn't count alone. And so that's important. I'm going to emphasize that right there. And so regulatory mechanisms can operate after transcription. And so what are we talking about? Well, it's so that the cell can sort of fine-tune gene expression in response to environmental conditions, so it's able to roll a little bit. And one of the ways it's able to do this, I foreshadowed this or alluded to it a little bit earlier, is that there's RNA splicing that takes place. And so from a particular messenger RNA transcript, certain segments of RNA can have certain introns removed and others not removed. And so that's important. Let me illustrate that with a little animation right here in terms of RNA processing. Okay, let's take a look at this.
A cell can rapidly adjust to environmental changes by regulating several processes that happen after transcription. In RNA processing, a cap and tail are added, introns removed, and the remaining exons spliced together. Alternative splicing of RNA may create different mRNA molecules from the same primary RNA transcript.
So there's alternatives in RNA processing. So in other words, the 5' cap and the poly A tail are consistent always, but it depends on how you're editing the messenger RNA, and you get some variation in terms of how you want to roll with post-transcriptional RNA processing. Okay, so I hope that was good. Okay, and now I want to sort of kick around this whole idea of RNA degradation. So in other words, the lifespan of RNA in the cytoplasm is important in determining protein synthesis. And so very simply, before we get more detailed, is that if messenger RNA is short-lived, then there won't be a lot of protein production. If it lives a little longer in the cytoplasm, then there'll be more translation, more protein. And so just as a rule of thumb, eukaryotic messenger RNA lives a lot longer than prokaryotic ones. But what's interesting about this is that there's a nucleotide detail, there's a nucleotide sequence that we've discovered that influences the lifespan of the messenger RNA, and it's in this sort of untranslated region. It's a residue called UTR, and it's at the 3' end of the molecule. Take a look over here at this video.
The lifespan of mRNA molecules helps determine how much of a protein is made. Nucleotide sequences that affect mRNA longevity are often found in the 3' untranslated region following the stop codon.
Right there.
Enzymes attack the poly-A tail first, and eventually the rest of the mRNA. Depending on its ability to resist this degradation, a eukaryotic mRNA molecule may last from hours to weeks. The longer an mRNA lasts, the more protein is made.
So that's kind of interesting. It's like you kind of want to get rid of it because or keep it around if it's useful. In other words, like you don't want to keep the newspaper around too long because it's old and you want to just recycle it. But it might be useful for a little while to keep it in case you want to refer to it. Okay, and so I also want to bring out another point. This is an interesting thing. The initiation of translation. So the initiation of translation of selected messenger RNAs can be at times regulated itself. So in other words, there's some proteins in the cytoplasm that can bind to messenger RNA and block it. And so therefore protein synthesis doesn't occur. We can also talk about the proteins themselves. And so we're sort of now, we're past transcription, we're past translation. What we're looking at is post-translation. So if I can emphasize this, so after translation, so the protein has already come off the ribosome. And so I have a little model here to show you. And so if you had a protein like this, a polypeptide chain coming off of the ribosome, you might say to yourself, well, it's three-dimensional conformations, probably ready to roll. I mean, it has a particular active site. If it's an enzyme, it's ready to go. But as it turns out, post-translational modification is important. Sometimes what the cell will do is attach particular chemicals to it. They'll attach a phosphate to it, and therefore the shape of the protein will be changed. And so you could modify proteins in a variety of different ways just by adding functional groups to it. And it's like this is a real frontier in biology these days, because it's not as simple as we may have thought that there's a gene and it makes a particular protein and there it is. But humans have a variety of enzymes that will add all sorts of functional groups that will further change the tertiary structure of a protein and therefore affect its functionality. Let me show you this, for example.
Often, polypeptides must be modified in some way to produce functional proteins. For example, a polypeptide is cleaved in 2 or more polypeptide chains. Other molecules might be attached to the protein, or the protein might be phosphorylated. Some proteins need to be transported to specific destinations.
Okay, what I want to emphasize with this is there's concern, for example, the enzymes necessary to attach a phosphate, the kinases, or to cleave a peptide. Some of those enzymes are, obviously they're found in eukaryotic cells, but if you're splicing in DNA into a prokaryotic cell from a eukaryote, sometimes prokaryotic cells lack the post-translational modification enzymes necessary for producing a functional protein. If we want to actually produce a protein through cloning and therefore purify it. So sometimes eukaryotic cells need to be used when transforming during DNA recombination, just saying. Okay, and then I find this to be interesting too in terms of protein regulation. Ultimately, sometimes proteins, when they're being produced, they need to be recycled too. When they're done, you need to like destroy them. So there's these proteasomes. Pretty cool. A proteasome is sort of like a trash, what's the analogy I'm thinking of? A disposal, where you're putting your stuff in there and it's just like grinding it and breaking it up into pieces. So these proteasomes are these giant protein complexes that can actually bind to protein molecules and degrade them. And so what's interesting is the cell marks the protein to be degraded by adding a molecule called ubiquitin. And then when the protein is ubiquinated, how's that for a word? Then it goes inside the proteasome and it's like just broken down into oligopeptides. So let's take a look at that. That's pretty cool too.
A cell can get rid of abnormal or damaged proteins and limit the lifetime of functional proteins by means of selective degradation. The cell marks the protein for destruction by attaching small ubiquitin proteins. Giant protein complexes called proteasomes recognize ubiquitin and break down the tagged protein.
And then those amino acids or peptides can be used for the cell for whatever purposes. It's like the cell's extremely cautious and conservative about not using too much energy and also recycling its molecules. And that's kind of the whole point of this video in terms of gene regulation. The cell wants to do certain things at certain times during development. It's involved in specialization. So pretty cool. The cells regulate gene expression in terms of transcription initiation, and we talked a little bit about RNA and some proteins as well for a little bonus. I hope you enjoyed something. Hope you enjoyed walking on the hill. Went up pretty far, but I hope you're still there with me, and thanks for watching.